from Christy Fleet as the advisor, Kaitlynn Stowers. Kaitlynn is a senior biology major. Her project was sparked by family members' experience with tobacco farming and COPD. Over the past 2 years, Kaitlynn has led the way to develop a project that is uniquely her own. She has spent countless hours in the lab running experiments, optimizing techniques, and troubleshooting to obtain the results she will share with us today. Her tenacity, leadership, and willingness to challenge ideas and ask difficult questions has led her to an intriguing result which suggests ways that nicotine exposure, independent of cigarette smoke, may affect our biochemistry and perhaps even our health. Kaitlynn's plans for next year include preparing to apply for PhD programs with a focus on biological or biomedical research.
Hello, I'm Kaitlynn Stowers, and I'm going to share with you guys my project, The Effects of Nicotine on the Expression of CHRNA3 in Correlation to Chronic Obstructive Pulmonary Disease. Or, more simply put, can simply nicotine cause you to have a chronic disease? I'd like to start out with what made me want to do this project to begin with. So growing up in Southwest Virginia, my family had a tobacco farm. For those of you who don't really know much about tobacco farming, it involves the entire family down from the 4-year-olds all the way up to the oldest generation. So my grandfather and the men of his generation were the predominant workers of this tobacco farm that we had. And as I grew older, I learned that all of them had been diagnosed with COPD. But I also knew that not all of them smoked tobacco products. Since tobacco smoking is one of the leading risk factors in the development of COPD, I raised the question, what caused the other ones that didn't smoke to develop this disease? So as I dug into the research, I learned that there could be a genetic impact or factor that led to them obtaining this disease. And I wanted to investigate more, which led me to my question, can nicotine cause a difference in the expression of a gene, specifically CHRNA3? So CHRNA3 is a gene that or more broadly put, something in your cells that sends messages to and from your brain or nervous system. And the expression of that gene is how well you interpret it. So let's say you went and bought a Lego kit to build a Lego spaceship. The instructions in this kit are your gene. How well you interpret them or how well you can build this ship is your expression. So typically the instructions make for you to build one Lego spaceship. So one Lego spaceship is your normal expression. If your expression is upregulated, that means you made more than one of these spaceships, then that's an upregulation. But if you don't make quite one or you make no of these spaceships, then you have a downregulation. So you typically want to keep your body at a normal expression level. So why might this be interesting to me? If we can say that just nicotine can cause a differentiation in your expression, then should we raise the question of how safe are nicotine products like nicotine gum or nicotine patches? Both products on the market help to cause people to quit smoking or quit using other tobacco products. Or how safe is that child who has parents that smoke but don't expose them to the secondhand smoke, but they're exposed to that nicotine residue when that's left over on that parent's skin when they pick up that child, and that child is then exposed to that nicotine. So that led to my hypothesis that nicotine might cause an upregulation in gene expression. So I've talked about COPD, and I'm sure some of you all are wondering, what the heck is COPD? What is she talking about? So COPD is an umbrella term for emphysema, chronic bronchitis, and various types of asthma. It is the third leading cause of death in the United States, so it wasn't consistent, which still made me question, can it just be nicotine that causes the development of COPD in someone? So I'd like to give a little recap on CHRNA3, which is the gene that I picked to study on since it is a major focus. Like I said, its predominant function is to send messages to your brain and your nervous system, but its secondary function is to bind nicotine. So it accepts nicotine, it wants to find nicotine in your body. And I picked this gene because a lot of research has said, like, hey, there might be some sort of correlation between this gene and someone having COPD. So for my research, I took mouse cells, which you can see right here, and in the food that these mouse cells use, I put various amounts of nicotine in it, and I exposed them to it. So I let them eat this nicotine-induced food for various hours. So anywhere from 1 hour to 48 hours, depending on where I was at in my project. And then I looked at how well they could grow. So did it make them grow larger or smaller? Did it kill them more? Like, what effect did it have on that? And after seeing that, I looked at how well these cells could interpret messages after the nicotine exposure. So were they able to interpret more or make more spaceships? Were they able to interpret less or make no spaceships? Or was it not affected at all and they just made that one Lego spaceship at the end of the day? So after 1 hour of exposure to this nicotine, I saw a very slight decrease in a very low concentration, but not of any significance. At a higher concentration of nicotine, I did see an upregulation. So they were able to make more spaceships than normally, which led me to look at that exposure at a longer time. So at 24, 36, and 48 hours, I saw a consistent upregulation. So these cells were able to make more spaceships, which was not necessarily a good thing because you only technically want one. But my data varied across the board as I was running it. So future analysis can decide and decipher whether or not this is consistent with just nicotine and it's not regular on its effects, or if it was just something in the project that needed more ironed out. So what did all this data mean? Like, why is it important? So since this gene has been said, like, hey, there's correlation between COPD and this way I'm expressed, and my data has shown that this gene can be upregulated, we can kind of make a connection between COPD and someone having this gene upregulated. So why does that even matter to anybody? That goes back to those questions I raised earlier. If we are able to say that just nicotine, not all the other chemicals in tobacco products, can cause someone to have COPD, should we consider looking at how safe nicotine gum is, nicotine patches, e-cigarettes, or other products that just have nicotine in them to our health and whether or not we can develop these chronic diseases? Or how safe is that child from the nicotine residue that's on their parents' clothes and body after they went out and smoked, but were not exposed to that secondhand smoke? So should we continue looking at nicotine by itself as a health factor and health risk for future generations? From my project alone, someone else can stem multiple other projects, so to continue looking at this benefit. So should someone look at how it is done in human cells, or what the effects of human versus mouse results could be? And to continue looking on to like other risk factors of COPD. What else can be known about this gene, since very little itself is known about it. But also, you can look at just nicotine's effects across other body parts, so other genes and other organizations in your body. I would like to end by thanking my advisors, Dr. Christine Fleet and Dr. Melissa Tabner, the biology department here at Emory & Henry, as well as the Ampersand Grant and Committee for the funding of this project.